And now we're going to spin around back to the United States, to New York City, to an old and dear friend of mine, Orrin Devinsky, who's the director of the NYU Langone Comprehensive Epilepsy Center, the NYU Grossman School of Medicine. He is the founder of FACES, Finding a Cure for Epilepsy and Seizures. In fact, I remember quite a few years ago now, because it was back in the World Trade Center, at an amazing event that you held for that gala. He's also been in Time magazine as one of the 50 health professionals changing the world, and here with us to speak about what is ahead for KCNQ2, particularly when it comes to gene therapies, which I know everyone has a lot of interest in. So Orrin, thank you so much for joining us via Zoom.
Thank you so much, Sarah, and appreciate the kind words, and it's great to be here with all of you. And I'm going to give somewhat of a whirlwind overview and tour of gene therapies for genetic epilepsies. It's a rapidly evolving and exciting field, although one still with many limitations. So just to begin, and I think the KCNQ2 community knows this well, there are reasons we need to make genetic diagnoses. I think you in the room know this better than anyone, but for example, we know sodium channel blockers are often effective, whereas in Dravet syndrome, those drugs are actually harmful. So having a genetic diagnosis can point us to dietary therapies, can point us towards specific meds, can point us away towards other medications. So kind of just as an overview, when I started out, people thought, why would you ever want to get a genetic diagnosis? You can't do anything. Well, of course, that changed radically, and it started with small molecules, and now it's evolved. So gene therapy was something of a potential during much of my career. There was a terrible case at University of Pennsylvania of a young man who died from a gene therapy. It put it on hold for probably a decade, but it has rebounded strongly, as well it should. And you can see these are some of the approved gene therapies as of 2019. Some gene replacements for neurologic disorders. You've heard about antisense oligonucleotides like Spinraza from the previous speaker. And of course, you can take a patient's own cells and modify them outside of their body, called ex vivo gene therapy. It's been used a lot in cancer for the chimeric T cell regulatory changes, but others as well. So it's a rapidly evolving field, and this list has already grown significantly since 2019. For those of us who deal with brain disorders, either as patients, parents, or scientists and physicians, we have a lot more challenges than the average people in the gene therapy field. If you're trying to treat the eye or the heart, you can just take a needle and put it into the eye or the pericardial sac, and you'll get great delivery. Mammals have a blood-brain barrier, so there's a reason why charged molecules, if you eat a poisonous mushroom, you may get very, very sick, and you have a liver to detoxify it, but that poisonous compound will not get into your brain. And it's one of the reasons we survive many things. That barrier is also a problem for getting therapies in. We have been able to design certain viral vectors, literally viral capsids, the protein that surround a DNA or RNA virus, that literally can be used to transmit genetic elements. These pose problems because our body recognizes these vectors as foreign and will make an immune response, often limiting therapy to a single dose. If you give it again, neutralizing antibodies will take out the vector and trigger an immune response and make you sick. So it's a double bad. The dosing for some of these, we really have to be careful. We want to get enough in to do what we want, but if we overdose, as many of the neurogenetic disorders, if you have an extra copy of a gene, you could end up with more problems as well. So many of the disorders are a lack of function, or one good copy and one bad copy, called haploinsufficiency, but too much of a good thing can be a problem as well. One of the other things we worry about with any genetic modification is what are called off-target effects. Are we going to modify normal genes or other parts of our genome, which I like to call the switches? We all know what lights are. They turn things on. Think of lights as genes, and think of regulatory elements or non-coding elements as the switches on your wall that turn genes on, turn the lights on and off, and dim them up and down. Those are pretty darn important, and we probably have many more of these regulatory switch elements than we do genes. Another issue is how long will the gene therapy work? For ASOs, they have to be redosed every 3 or 6 months. And then there may be counter-regulatory effects. Biology is wonderfully adaptive. It's had literally billions of years to figure out what it does. And we try to muck around in the system, and what we do may work to some degree, but there may be counter-regulatory forces that we need to consider. This is a slide from a recent article by Gemma Carville at Northwestern, and it just looks at some of the broad types of gene disorders. I'm not going to spend much time, but keep in mind for KCNQ2, typically we're dealing with a haploinsufficiency or a partial loss of function in one copy of what should be 2 good copies of a gene. So this slide summarizes the broad spectrum of approaches that are out there for gene therapy. The first is small molecules or transfer RNAs, which are one of the genetic elements, to overcome premature stop codons. So premature stop codons, I'll get to in the next slide, but they represent a very substantial portion of causes of genetic diseases. And one of the nice things about premature stop codons is it probably represents, give or take, 15% of KCNQ2 mutations that are causing disease. And this is where the KCNQ2 community shares features with Dravet, with SCN2A, 8A, with every other neurogenetic and many non-neurogenetic disorders. So it's an area for collaboration. Antisense oligonucleotides you've heard about, they're already in the clinic for Dravet syndrome. I mentioned the transfer RNAs. They're a way to do both read-through and overcome a premature stop codon, also to increase the messenger RNA production or half-life and thereby increase protein production directly. I mentioned the switches on the wall for the lights. Those are regulatory elements. Those can be identified for every gene, including KCNQ2. They're far smaller than the gene, so they're much easier to package into a viral vector or a lipid nanoparticle and deliver. CRISPR gene editing, I will talk about some efforts we've had in our group to do some gene editing on KCNQ2 mutations, but you can correct a single letter change, you can correct a missing letter or two or an additional letter or two. There's a lot of opportunities to do that. And these work fantastically well in a Petri dish, and they can work well in mice if you inject them into the brain. Getting these delivered to the brain remains a problem. So there are many multi-billion-dollar CRISPR companies that are out there. None of them are currently focused on central nervous system disorders because of the delivery challenge. There's epigenetic editing, which can do many things, but a lot of the focus for brain disorders probably not relevant to KCNQ2, as it's good for X reactivation for diseases like Fragile X, Rett syndrome, CDKL5, and then gene replacement therapy, just getting a whole copy of the gene put in. So this slide goes over premature stop codons just briefly. As many of you remember from high school biology, there are 4 letters, C, G, T, and A. Each codon, each 3 letters of nucleotides codes for 1 amino acid. So back in the 50s, when Watson and Crick were figuring this out, and then later they realized there are 3 nucleotides, DNA letters, for every amino acid that's coded, it then became clear, well, there's 4 letters at each spot, so 4 times 4 times 4 is 64. We only have 20 amino acids. They literally figured that out at the Eagle Pub outside of Cambridge. And we're wondering what the heck all these other letters were doing, and there's duplication. Evolution was very smart. So of the 64, 61 code for 20 amino acids. The amino acids, the simple ones like glycine that probably came about in evolution very early in life's history, can have up to 8 different combinations, while those that came much later, like a methionine, have only 2 or 3. Of the 64 combos, 3 code for stop. And that's literally like you're producing a shirt and the sleeve length should be 20 inches. And at the end of the 20 inches, one of the 3 stop codons. However, if that one of those letters is changed and an amino acid, let's just say glycine, one of its codons has a change and it now says stop, you'll have a sleeve length of 10 inches instead of 20. The cell will recognize that that messenger RNA is quite abnormal and digest it before it typically gets out of the nucleus. So you will lose half the gene because of those stop codons. And as I said, these represent 12% of the genes causing human inherited disorders. and about 20% of single base pair changes that alter the coding regions of genes. And there are small molecules, gentamicin, which is an old-fashioned antibiotic, is one of them, that actually can read through the premature stop codons. And this certainly is an avenue of research and therapy for cases that in KCNQ2, like I said, I don't know the number, but I'm guessing about 15%. There are some other drugs, and there's at least one company, Tavard, out of Cambridge, Massachusetts, that was founded by two dads with children with Dravet syndrome. It's actually Dravet spelled backwards. That has a very effective suppressor tRNA that stops these premature stop codons, so again, allows read-through of the proteins. I'm next going to turn to the antisense oligonucleotides. You've heard a little bit about those. These are out there, so these are exciting. These can be done, and I'll talk about one company that already has a trial in Dravet syndrome. These are single strand of nucleic acids. They're synthetic and they're modified. The modifications allow them to prevent digestion. Normally, if you just took a string of oligonucleotides, oligo just means a small number of nucleotides, and put them into the spinal fluid in a lumbar puncture, which is where they're typically delivered, they would get digested within hours, if not days. The modifications are made allow them to last for months, and they seek to correct the dose of messenger RNA transcript from a disease-causing gene, and as you heard, can already be used. So this just looks at again, an overview of some of the major ones, and you can just look at the bottom there. You can exclude a bad exon, as has been done in spinal muscular atrophy. You can exclude what's called a cryptic exon that can mess things up with a stop codon, as in Batten's disease. Or you could exclude what's called a poison exon. That is the approach that Stoke Therapeutics is using in Dravet, where there's actually a premature stop codon that kind of interrupts the whole sequence. So you just splice away from that extra piece, and the messenger RNA produces a protein that works just fine. So kind of a very clever approach. But basically, these ASOs can be used in the examples here to increase gene expression, to downregulate gene expression by different mechanisms. So this is the TANGO, or targeted approach to increase the messenger RNA in the SCN1A gene that produces the NaV1.1 sodium channel protein. And essentially what it does is a group of oligonucleotides, nucleotides bind to that site and prevent that codon from being transcribed. And that codon, when it's part of the pre-messenger RNA, will lead to degradation of that pre-messenger RNA and essentially not get it to be produced. So the goal here, again, just to step back, because it's a little complicated, and for all of us in the business, it took a while to understand. Keep in mind, the Dravet is typically a haploinsufficiency, which is true for many KCNQ2 disorders as well. So you have one good copy and one bad copy. This ASO will increase the production of both good and bad copies. We don't care about producing the bad copies because they don't have any negative effect. They just don't work. They get digested either in the nucleus or the cytoplasm, and they just don't form functional proteins. If you can double the amount of the good copy of the gene, you get back to a normal state. And indeed, they have shown in animal models. Here, this is a single intracerebral ventricular injection of their antisense oligonucleotide meant to avoid that poison codon with the premature stop in it. And if it's given postnatal day 2, it reduces both seizures and the SUDEP rate. These mice, and unfortunately the human patients with Dravet, have very high rates of SUDEP, and can even work at postnatal day 14 as well. So one of the things we look for, it's easy to fix mice at postnatal day 1, but obviously for most of you in the audience, you know that your children are not diagnosed on the first day of life. It takes time. So we want to find therapies that can be effective later in a child's life as well. But I think most of the gene therapies will be helpful. I don't think any of them, for many of the developmental and epileptic encephalopathies like KCNQ2, are going to fully reverse the disorder, whether the whole gene is replaced, whether it's an ASO. But I do believe strongly that they can very much normalize the function of nerve cells, allow seizures to be better controlled, and probably improve fine and gross motor skills as well as cognitive and behavioral function. And so I think there's a lot of upside to gene therapy. It's just not going to be a cure for a 5-year-old who has, severe developmental delays. And this is another slide from a recent Science article, just to show that these ASOs can be used to decrease pathogenic proteins. So that there are many neurologic disorders where the protein is abnormal, it folds abnormal, it leads to accumulations in nerve cells that kill the nerve cells, a lot of the progressive myoclonic epilepsies, some of the neurodegenerative diseases like Parkinson's and Alzheimer's and frontotemporal dementia may have these mechanisms. So this is yet another way that ASOs can be used, but not really relevant to the KCNQ2 community. So this slide summarizes an approach with regulatory elements that I believe will be an important one in the coming years. It's not quite there yet, although one company, Encoded, has a regulatory element approach using zinc fingers to treat Dravet syndrome, and that hopefully will get to the clinic within a year. But there are 516 recessive disease-causing and 660 haploinsufficiency disease-causing genes. And a very significant number of those, about 1/4 of the recessives and about 1/5 of the haploinsufficiency genes are too big to be packaged into an adeno-associated virus, which is essentially the main way that people are delivering gene therapies into the brain at this time. So you need those adeno-associated viruses essentially can carry 4,700 nucleotides or base pairs. 1,700 have to be used to drive the expression. There's promoters, there's enhancers, things like that just have to be in there. So we're limited to genes with 3,000 base pairs or less if we're going to do whole gene replacement. The nice part of the regulatory elements is they tend to be much, much smaller, in the order sometimes of 20 or 40 base pairs. So they're tiny compared to genes that may have 2,000, 3,000, 5,000 base pairs in them. However, the regulatory element approach that's been used is either a zinc finger, which is a little smaller. This is one that uses what's called a deactivated Cas9. As I'll tell you, Cas9 is the bacterial enzyme underlying most of the CRISPR biology that's been reported in the press and used in treating human disease. What's beautiful about the Cas9 is it is exquisite in localizing parts of the genome when attached to a single guide RNA. And you can see the sgRNA below, which is about 14 to 22 base pairs. It recognizes an exact sequence in the genome. And that's what's amazing about it. You just need 14 to 22 letters, and within the 3 billion letters in our genome, you can get to the exact spot. And then depending on which DCAS it is and regulatory element, you can put a regulatory element in that increases the expression of the gene by binding to the promoter site, or it decreases it by inhibiting the promoter site. So the DCAS9 is really your navigation tool when it's combined with the single guide RNA, and then the regulatory element can be an enhancer or an inhibitor of that promoter to turn the gene on or off. So again, same thing here. You have a haploinsufficiency like KCNQ2. You get the regulatory element identified, you package it into an AAV with this DCAS9 and a single guide arm, and you can double the expression of the good copy of the gene. And this has been done to date in animal models and cured disorders like a SIM1, which causes obesity. It's a gene expressed in the hypothalamus, and there's already been cures in mice. It's been used in SCN1A, as I mentioned, and SCN2A as well. So I think you'll see more of this in the future, but I don't think it's quite ready for prime time with KCNQ2, but would be a worthwhile place for the foundation to think about putting time into. So this is Encoded Therapeutics, which I did mention. They have a platform that optimizes the regulatory elements. It can identify them. And then we'll use a zinc finger packaged into the adeno-associated vector. And it can last up to 10 years in non-dividing cells like neurons. So really quite a beautiful approach. It certainly works well in the Petri dish. It works well in mice. And hopefully in 2023, we'll start to get an idea of whether it's working in humans. And this just shows that with regulatory elements, not only can you turn genes on and off, but you can turn them on in very specific and precise groups of cells. So for Dravet syndrome, which is where they're focusing, the cell type of interest is an inhibitory interneuron, which make up a minority of all our nerve cells in our brain, but not just any inhibitory interneuron, it's the parvalbumin positive, which is a subgroup. So it's a tiny percentage of the cells they want to target, and you can see on the right how selective it can be for a really tiny portion of the nerve cells in that animal brain. And as I mentioned, regulatory elements can be used to drive gene expression up or down. I think for KCNQ2 and the vast majority of neurogenetic epilepsies, we're looking at increasing a haploinsufficiency state. So this is, again, some early work that they've done. They use intracerebral ventricular injections, and it's expressed in the GABAergic interneurons. And similar to what we saw with Stoke, down here you can see the SCN1A messenger RNA is increased, the NaV1.1 protein is increased, and the effect is to decrease seizures in these Dravet mice that are induced by heat or thermic stress, and also decrease the frequency and severity of spontaneous seizures. This is another company with which I have some involvement. So just on the disclosure side, this is Regal Therapeutics. They've also looked at SCN1A and have a slightly different approach on the regulatory element side. But this is from a Nature Neuroscience paper now 3 years ago. They verified that they can also exquisitely selectively turn NaV1.1 or SCN1A gene expression in parvalbumin positive cells in the rat, in 2 species of monkeys, the marmoset and the macaque that you see on the upper left, and also in human cells taken from patients who had epilepsy surgery, and that's on the far right. So it's really a quite beautiful system, these regulatory elements. They can be super selective, and they can be tuned pretty well. And for many of these systems, fortunately, the cells have kind of a homeostatic balancing system, and if the gene gets expressed too much, there are endogenous or internal mechanisms to kind of dampen it down and get it to the right level. So the next broad area I'd like to touch base is CRISPR editing, and this has received a lot of input in the press. It's a Nobel Prize was recently given to Jennifer Doudna and colleagues, and a book on her was written, a bestseller on her life. So these are DNA sequences in bacterial genomes. They're basically, they take fragments of viruses, bacteriophages that infect bacteria, and they use them to identify the bacterial genomes and splice them apart. So essentially, it's a way to combat viruses by bacteria. I like to say, you know, if you get a textbook of biochemistry by someone like Leninger, which is what I used anyway when I was in college and medical school, that textbook was written by bacteria. Everything pretty much in biochemistry, 80% of it, all the synthetic pathways of compounds, amino acids, making DNA, copying DNA, bacteria did that all. They invented CRISPR. We give a Nobel Prize away for discovering what bacteria did. It would be like someone digging up a wheel in Mesopotamia and saying, I get a Nobel Prize for finding this wheel. Someone invented the wheel 4,000 years ago. But it's really quite remarkable what the bacteria have done and what we've been able to take. So these enzymes can be used to very precisely edit animal or human genes, correct mutations, skip abnormal exons, many, many things. And as I said, there are many companies currently active that are in trials and that have gotten approved therapies, especially the ex vivo ones for blood disorders. Those are very easy to access targets, liver, heart, things like that. And that's where these CRISPR companies are currently focused. So this is a child I care for with a KCNQ2 mutation, and my colleague Chenzhu Long at NYU has used an adenine base editor. This, I think, was first really reported by David Liu's group at Harvard and the Broad, but it's a modification of his Cas9. This is not a dead Cas. This is a live Cas. This is an enzyme that actually does edit DNA, and you can see in this slide there was a mutation that turned a G to an A, and he's able to restore it back to the normal letter. So it's a single letter change in exon 6 of this KCNQ2, and he's able to get it back. I should say it was converted from a normal G to an A, and he's able to get it back to where it started. So again, a very remarkable system. The challenge here, Chen Zhu was able to do this, Dr. Long was able to do this probably in like 3-4 months in a Petri dish, as these CRISPR editing people are geniuses in ways I don't understand, but getting this into the brain is a big challenge. And again, this just shows actually showing the proteins and looking the DNA sequence, but it's essentially a proof that indeed in these cells, he has been able to correct that mutation back to the normal 3-letter codon. And so what are the challenges in bringing CRISPR to the brain? As I said, there are many multi-billion-dollar companies, but none have even a brain disorder in their pipeline that I'm aware of. And the reason is delivery. We have 90 billion or 100 billion nerve cells in our brain. One question is how many have to be repaired? And the bigger question is how do you get CRISPR in? It's a big molecule. It can stimulate an immune response. The adeno-associated virus 9 can have toxicity. It's given via the spinal fluid to the dorsal root ganglion, if it's given intravenously to the liver. So one approach that's being used now, not so much for CRISPR, but for gene replacement, is a direct injection into the spinal fluid. And what you see here on the left side is an intrathecal lumbar puncture. That's a standard LP. Any neurologist has done this hundreds of times. It's a routine procedure in emergency rooms if you're worried about meningitis or encephalitis. And it's also a way to deliver drugs or therapies. And in this case, ASOs are currently being used in the Stoke trial via that intrathecal lumbar puncture. To get something to the brain in high doses, however, the approach that's now being used is to inject it into the very base of the brain over here. There's a big fluid space right at the very bottom of the brain, kind of where the brain and spinal cord meet, called the cisterna magna. Cisterna is just the Latin word for a cistern or a container of fluid. Magna means big, so it's the big cistern in the brain. And it can easily be targeted and gives widespread distribution in the brain. And I believe there are a few gene therapy replacements, not CRISPR, but gene therapy replacements that use this approach. And Ultragenyx is currently using this approach in a design trial that we hope will go live in something like early 2024 for CDKL5 as a full gene replacement. So this slide just looks at some of the diseases on the top, I think 6 or 7, where you actually can replace a gene, just replace the entire gene. This could be done potentially in the brain, but it's being also done certainly with the liver and other places. And it's a way to increase the expression if you have a haploinsufficiency. As I mentioned before, some of the neurodegenerative diseases are caused by bad proteins. In those cases, you want to silence it. And it's also been used for that function as well. And this just gives a little bit of the molecular biology of how these gene replacements are here, but it's really, you need to get the promoter region. The transgene is the actual gene itself. And that's the part that you want to get transcribed and made into a protein. So as we finish, I'm going to leave you with challenges because we still have a lot. We have a blood-brain barrier that only likes lipid materials, not charged ones. We have to get through the cell membrane. Can we use condensed DNA? It's got to be small enough to be absorbed by the nerve cell. It's got to escape from digestive forces in the cytoplasm. It's got to get translocated across the cytoplasm. Often active mechanisms need to be used. It has to enter the nucleus. It's got to get into the genome or sit nearby and get expressed. And then you want it ideally as a regulatory element or a gene replacement to get expressed for a very long time. And we're fortunate that the nerve cells are not dividing. So that is something in our favor. But gene therapy does face problems. The promise is that, as I said, we have ASOs that are in the clinic, regulatory elements, hopefully we'll get there soon, gene replacements will get there. And I'm optimistic many of the other modalities like transfer RNAs and CRISPR will get there as well. So thank you very much and happy to answer a few questions if there are any.
Thank you. Thank you so much, Warren. I hope you're hearing the applause. We have more questions than this, but if we have any additional ones, we'll need to ask them online. But the one question that I'm getting from more than one person is, and I know it's tricky because you've talked about a number of different therapies, but can you give people any sense of a timeline on this? And I know that's hard.
I think part of that answer may depend on the KCNQ2 community a little bit, how much you want to invest. I can tell you, I just came from a 2-day meeting in Cambridge this week from the Lulu Foundation, which is a family from Dubai whose daughter I've been involved with her care. It's a wonderful family. They probably had 200 people in the room. They had Billy Dunn, who's the head of the neurology branch of the FDA. They had 8 companies in the room. There's already a small molecule approved that they stimulated the testing for, ganaxolone from Marinus, but there's trials ongoing with small molecules from UCB Pharma with fenfluramine, with Takeda's Solistat, and they also were the ones who stimulated Ultragenics, and they have 2 or 3 other gene therapy companies working on this as well. So I would put it back to you guys. I think you need to get together and make this a priority. It's not easy, it's expensive, but you need to make it an easy thing for companies to want to go into. And I think that's what Lean and Majid Jafar have done and have succeeded spectacularly. Like I said, the Ultragenics gene replacement will probably be in the clinic in 14, 15 months. There's already an approved drug. When they had their first meeting 7 or 8 years ago, I was at it, and Majid said, I want treatments within 5 years. I want cures within 10. He's on target to get both.
Well, that's exciting, and I think it's a call to action for the community. Following up from that, I'm assuming you think people, well, I shouldn't assume. Do you think people should participate in things like registries, Citizen, those kinds of things? Is that helpful for the development of moving things along?
Absolutely. If you're a company, and I listen, I speak to companies all the time, and they literally, they have a regulatory element, they have an ASO. Like Oren, there's 40, there's 80, there's 120 neurogenetic disorders. Which one should we focus on? And they literally have boxes that they go through. How many people are they? They don't want it to be too rare. They don't want it to be too common. It's not an orphan disease. They lose some of the credits for that. But one of the big boxes is there an efficient organization that has patient registries? So if they want to know how many people have premature, what if a company said we have a great drug for premature stop codon? It's a small molecule, it's a piece of cake relative to a gene therapy, but it does modify the gene. Can KCNQ2 tell us how many people have premature stop codons? You want to be able to just go on a database, punch in a few things, and get that answer in a minute. There's no reason you can't do that.
All right, thank you so much. Really appreciate it. Again, round of applause for Dr. Davinsky.
My pleasure. Take care, everybody.